All right, everybody. Hello. How are you feeling about life?
It's Friday. I know it's Friday.
It is Friday, so that is a good thing. Feeling like you're surviving? Ish. I'm feeling like this spring term's kicking my butt, frankly, but I'm like, you have. It's okay if you don't have a lot of sympathy for that, right? I'm not complaining. This has been fun.
I'm taking 7.5 credits.
What?
I'm taking 7.5 credits right now.
7 point. Oh, so this isn't the only thing you're doing.
No. I can hear you yapping right.
All right. That is tough. Well,
So now you know I hate that.
now I know. So some of you may have had some questions about, for example, it's a 4 credit hour class, but we're meeting every day. What's up with that? I thought a 4 credit hour class was supposed to meet 4 times a week. The answer to that is that usually for our classes, our 3 credit classes meet 3 times a week, and then there's a optional but highly encouraged lab review session once a week that most people go to. For 285, they decided not to have the optional review, but to build in once a week sort of review. What we do in this class is we do review all along. So the time that we spend looking at quizzes, practicing problems in class is sort of the equivalent of that 1 hour a week, not necessarily new content, but just practice. So I'm trying to space it out there. That being said, I understand it feels like you're drinking out of a fire hose, and I'm sorry. All right. Nevertheless, the important part of what we're doing this semester is to try to give you a feel for what's going on around you at the molecular level, including outside you and inside you. And all of these reactions that we've been talking about are happening inside you right now. And the beautiful thing is that they all follow a similar pattern. So the pattern that we've been talking most about lately is addition of a nucleophile, which I'm always highlighting in purple, to either a carbonyl carbon or a phosphoryl group. You break the pi bond, you make an intermediate where the oxygen that's pink is negatively charged. And the other thing that blue phosphoryl or carbonyl group is attached to is a leaving group. So nucleophile attacks, you get a tetrahedral intermediate. We've talked about how to look at intermediates and come up with a decent hypothesis for how you get there. Then the electrons kick down and kick off the leaving group that I always highlight in green. You make a new bond. So the 2 new bonds that you make during this process are first the new bond between the nucleophile and the phosphorus, and then you restore the pi bond as the leaving group leaves. So in the first step, you make a new sigma bond and break a pi bond, and in the second step, you restore the pi bond and break an old sigma bond, and the leaving group leaves. And that's the pattern of addition to get an intermediate, and then an elimination to remove a leaving group. So those are our key addition and elimination steps, and then we've used proton transfer steps to grease the wheels, as it were, sometimes to make something a good enough nucleophile to attack, sometimes to make something a good enough leaving group to leave. And so I think if you look at that mechanism handout, I've posted a key that shows how to do all of the reactions on the handout you can look at. I think last time we didn't do thioester hydrolysis, we didn't do this last one, and we also didn't do pyrophosphate hydrolysis. And if you go look at the key, you should be able to identify each step as either addition, elimination, or proton transfer. Questions you have on things that aren't making sense? Go ahead, I'm sorry. I did start the recording, I believe. Yes, we're live. No, that's perfect. Okay, other things that are not making sense that you want to ask about? Yes?
We had memorized the terms associated with these reactions, and it's to provide like biological context.
Memorize the terms or,
You need to go into the exam knowing what.
these words, substrate level of phosphorylation. No, that's biological context. And I'll refer to this later. In fact, a lot of these in the next week, we're going to talk about metabolism of carbohydrates, and you'll have seen a decent number of these already in the reactions that we've talked about. In my regular OChem classes, we talk about reactions that you do in the lab, and we only get to the biological stuff later. But because in this class what we care about is biology, we're starting with the biological examples. That creates a problem because the biological molecules are much more complicated in general than what we would do in the lab. So you have to develop eyes to see past the non-essential details to the things that matter. That's why I'm using color and highlighting so much. And with practice, you'll be able to see it too. You'll know what to ignore and what to focus on. Yeah?
Is direct substitution just addition and elimination together?
So good question. And that direct substitution shows up on your problem set. I mentioned this, let's see, when did that happen? That may have been Tuesday. No. Yes? Still no. Trying. Wow, what a week. I'm trying to remember when we last talked about that. Nope. All right. That's okay. I'll just, instead of trying to find it, we'll just talk about it now. So you're most familiar with addition, elimination, and proton transfer. There is something that's mentioned on your problem set, and that I believe I mentioned in class at some point in the last few days, but this is the idea of a direct substitution reaction. There's not a lot of these in biology. Maybe it was just last time that I said this, but there's not a lot of examples of this in biology, but I'm just distracted by the fact that this page doesn't have our traditional grid. It's sad, but oh well. Yeah, I'm gonna fix it. And it doesn't still want to be fixed, oh well. Huh. Well, that's frustrating and sad, but somehow life goes on, doesn't it? Direct substitution is where you have an sp3 hybridized carbon attached to a leaving group. And there's no carbonyl. The LUMO, if the carbon leaving group bond is weak, then that means the carbon leaving group sigma star is low in energy, meaning it's the LUMO. Let's see. I am sorry, I actually can't handle the no grid. We gotta just start again. All right. All right, there we go. So here's your sp3 hybridized carbon. The carbon leaving group bond is weak, and therefore the LUMO is the carbon leaving group sigma star, which is low in energy. And what happens in direct substitution, unlike nucleophilic acyl substitution, in direct substitution there's not an addition then an elimination step in separate steps. In direct substitution, the nucleophile attacks the sigma star. There is no intermediate. You break the sigma bond, and then you make a new sigma bond between the nucleophile and the leaving group. In some ways, direct substitution is the carbon version of proton transfer. You know, in a proton transfer, the base attacks the hydrogen. Well, in proton transfer, whatever the hydrogen's bonded to, the base attacks the sigma star between that atom and hydrogen, and you break the hydrogen conjugate base bond. Direct substitution is like that. The leaving group leaves with its negative charge, and there is no intermediate. There's one example of a direct substitution reaction on your problem set. What you'll do to solve that is you'll look at the starting materials versus the products, and you'll try to identify what the leaving group is, and hint, it's a huge, large, unreasonably large molecule. The carbon that gets attacked is pretty small. Compare the starting materials with the intermediates and the products to see what's changed, but then understand that because it's a direct substitution, your nucleophile, whatever it is going to be dumping electrons into the sigma star for the bond between the carbon and the leaving group. Okay, follow-up questions on that? No? Okay. So we don't see a ton of examples of that. It does occur a little bit in biochemistry. I've mentioned it here for completeness. It's not necessarily as important as the addition and elimination steps. Other questions from last time, things you thought about that aren't making a whole lot of sense? How do I know how to do whatever? Yeah.
Our problem set mentions, there's like a whole question on hydrolysis reactions.
Uh-huh.
Have you gone over that yet? Or we've gone over that?
Hydrolysis reactions, yeah. So an example of a hydrolysis reaction is this saponification reaction. I think you're talking about the first problem on the problem set, which is like, second question? Oh, what do I know? Do I even have that here? Maybe. Let me just open it up. Huh. Oops. All right, is second question? That's right, this one? Yeah. So this question talks about hydrolysis of a triglyceride. I'll just send this to Notability so I can use a pointer. This problem talks about hydrolysis of a triglyceride and a small protein. This is a triglyceride. This is a small protein or a peptide. Models for clumps of oil and hair in your drains. You got a clogged drain. What's in there is a lot of hair and a lot of grease. And if you go by Drano to unclog the clog, whatever. Drano is just, you don't want to touch that or drink it. It's concentrated sodium hydroxide or lye. And what it's doing is it's breaking these molecules up into smaller pieces. And the hydrolysis reaction you've actually seen before in the mechanism handout. We didn't call it a hydrolysis reaction, but the mechanism is basically the same. So the reaction was saponification. That's ester hydrolysis. So what do? You have lone pair electrons on the OH minus, which is your nucleophile, attack the carbonyl carbon. You get a tetrahedral intermediate, and then the alcohol gets kicked off, and you're left with a fatty acid and a glycerol. So that's how esters get hydrolyzed, and that would be the same reaction that would occur with Drano and a greasy molecule within this clog. Now, notice that once glycerol comes off, glycerol should be perfectly soluble in water. It's plenty polar. And then you learned just earlier this week or last week about fatty acids in water. They're okay in water, right? Why? Remember what they form? Micelles, soap, right? So the saponification reaction will turn the grease that's in your drain into soap, which is great, and then it just washes away. Now the jump that you haven't seen, but we're asking you to sort of extrapolate is, what about amide bonds? Okay, amides are less reactive than esters. For an ester, what you have is a negatively charged oxygen in the hydrolysis reaction attacking one of the carbonyl carbons. You get a tetrahedral intermediate, and then the leaving group gets kicked off. By the way, there's 3 esters in this molecule, so this would happen 3 times to completely hydrolyze the triglyceride. What might happen here involves hydroxide as a nucleophile attacking the amide carbonyl carbon to get a tetrahedral intermediate. Let's just look briefly at what that tetrahedral intermediate is. Here's an OH, and here's an NH. We'll call this R prime. Now, as it stands, OH was the nucleophile, and NHR was whatever was attached to the carbonyl carbon, and this is the oxygen. What do you think about that as a leaving group? Is that a great leaving group, or is that on our list of things that are pretty not great? Pretty not great, right? RNH minus has a conjugate acid that's the neutral amine whose pKa is 38. So this is a really cruddy leaving group, but if nature were able to protonate it first, it could become a good leaving group. And then if it leaves, what are you left with? On one side, you have a carboxylic acid, and on the other side, you have an amine. Yeah?
Is it safe to say the higher the pKa, the better the leaving group is, or does it depend on the pH?
The lower the pKa of the conjugate acid of the leaving group, the better the leaving group is.
Okay.
The higher the acidity is. The higher the pKa of the conjugate acid of the leaving group, the worse of a leaving group it is.
Okay.
Yeah. So notice that when you do this hydrolysis reaction, you make a carboxylic acid. That may actually be the thing that protonates the nitrogen in this step. A carboxylic acid from another hydrolysis reaction protonates the nitrogen. In any case, as soon as you form the carboxylic acid, remember that we're in pure sodium, like very concentrated sodium hydroxide. So likely the last molecule of sodium, one of the molecules of sodium hydroxide deprotonates your carboxylic acid, and you're left with a carboxylate. So that's an amide hydrolysis reaction. But even though the players are similar or are different, you can see the similarity to the ester hydrolysis reaction. The only issue is amides are tougher to hydrolyze than esters. So this is why you kind of have to wait a while. The instructions say you pour the Drano on the clog and you let it sit for a while. I think the esters hydrolyze pretty quickly, but the amides in the hair do not. And it takes them a while. And eventually, as the hair and the grease begin to dissolve, then the clog isn't so cloggy anymore. And as things dissolve, then it can be washed away. Questions about that? Okay, other things that aren't making sense? You're scared to ask, because if you do, you're afraid I'll tell you something new that you have to know. Well, my friends, there's nothing that can prevent that now. Yes, go ahead.
So referring back to this question, so it would break the bond from the carbonyl group to the nitrogen, right?
Yes.
So then that would make it a lone pair living on the nitrogen.
Yep,
And then it's just like basically a domino effect, correct?
that's right. So in all of these cases, you have in each of in the triglyceride, you got 3 carbonyl carbons and 3 carbonyl oxygens. That is 3 places where the hydrolysis or attack of sodium hydroxide can occur. Similarly, in this protein or peptide, you have 3 carbonyl carbons that can be attacked and 3 carbonyl oxygens that can pick up the negative charge in the intermediate. And so, yeah, you do one of them and then the other and then the other in any order. Yeah.
So how come you don't use a water molecule instead of hydrogen?
Oh, to protonate the amine?
Yes.
Or to do what?
Like to bridge the bonds. Like how come?
Why do you use hydroxide instead of water to attack the carbonyl carbon?
Yes.
It's a good question. If you compare the stability of water versus hydroxide, hydroxide is more reactive, less stable because of its negative charge. So even though there is water present, hydroxide's a better nucleophile. Negative charge means its electrons are higher in energy. Yeah?
Wouldn't there be 5 different, I guess, reactions because there's still the other 2?
I'm sorry, there are, let's see, I highlighted the wrong things. Here are the, yep, and sorry, you were right because I was hurrying and not paying close enough attention. So let me highlight all of the amide bonds that are present in this molecule. I think it ends up being 4. This one, this is an ammonium group. This one's not involved in an amide. This is a carboxylate. This is also not involved in an amide. So there are 1, 2, 3, 4 amides to break in this molecule. Okay, what else? Yeah.
Would you want us to show the nitrogen being protonated?
So that's an excellent question, and the simplest way to do this is to not bother with it. I'm just struggling to justify that because what some people claim happens is this: even though the nitrogen's a cruddy leaving group, they say, it's okay for it to leave because as soon as it leaves as negatively charged NHR, even before it's really gotten out of town, it just steals a proton from the carboxylic acid that just was made. And then what you're left with is the carboxylate and the amide. That is probably okay. What I'm telling you is our NH minus is such a cruddy leaving group that it's got to pick up a proton either before or while it's leaving, probably not after. So if you wanted to show it this way, where you show a water molecule protonating that would be fine. If you wanted to do it this way, where the RNH minus leaving group leaves and then gets protonated by the carboxylic acid, that's the product of this step, that's fine too. Yeah, good question. What else? Yeah.
For like the triglyceride part, would the negatively charged ester always take the nitrogen from the carboxylic acid?
Would the negatively charged alcohol that's leaving always take the proton from the carboxylic acid like we saw in this saponification reaction? Yeah. Yep, because the alkoxide as it's leaving is a strong enough base to deprotonate the carboxylic acid. If you compare the pKa of the acid versus the pKa of this base's conjugate acid, which is an alcohol, which is 16, that tells you that that happens very quickly. Okay, others? Yeah.
So for the NH2, if it's in a pH of 7, would it become NH3?
Yes, at pH 7 it would become NH3. Good. Probably not in the clog itself because the concentrated sodium hydroxide is probably pH, I don't know, 11, 12 maybe. All right, others? All right. Well, so aside from these nucleophilic acyl substitution reactions, that is addition, tetrahedral intermediate, elimination, there are other interesting reactions that are important to biology that also rely on similar steps. And so I want to talk about some of these and work through their mechanisms and then give you a chance to do something similar, just as a preview for how nature will do things. So one new reaction is shown here. It's called the aldol reaction. And you have one of these on your mechanism handout, though it's not this simple version. I meant to revise that and didn't, so I'm sorry. You'll have access to this in the notes that I will post. In the aldol reaction, what's interesting is you start out with a carbonyl compound where the LUMO is the carbon oxygen pi star. So typical electrophile. But for your nucleophile in the aldol reaction, what you're going to do first is deprotonate this second carbonyl compound at the alpha carbon, and your goal will be to make an enolate nucleophile. We've talked about this kind of deprotonation before. We've talked about why the proton that is adjacent to or alpha to a carbonyl compound is somewhat more acidic, well actually extremely more acidic than you would expect for an sp3 hybridized carbon-hydrogen bond. Its pKa is around 20. And we've talked about the reason for that is that the conjugate base, after you remove the alpha proton, is stabilized by resonance. Here's the conjugate base. We call it an enolate, and you can draw a resonance structure where you take these electrons on the alpha carbon, you kick them down to form a carbon-carbon pi bond with what used to be the carbonyl carbon, and then the carbonyl oxygen gets the negative charge. This is an enolate, and enolates are nucleophiles. The negative charge is shared between the alpha carbon and the oxygen, but when enolates act as nucleophiles, it's mostly the alpha carbon that does the attacking. So what we're going to do now is simply standard carbonyl type chemistry, only we're using a nucleophile that you really haven't seen be a nucleophile before. So what happens first in this reaction is we make the enolate. Now, you can do that like I showed you up here. You can make the enolate, then draw a little arrow, and then draw the enolate. But if you're in a hurry, and in some biological mechanisms, the other thing you can do that's totally fine is to say, okay, the base removes this proton, that's going to turn this into an enolate, and the resulting negatively charged carbon is going to attack the carbonyl carbon. So you are welcome to do the deprotonation of the alpha carbon to get the enolate first in a separate step, and then show the enolate attacking the carbonyl like this. Alternatively, if you're in a hurry and don't want to redraw a bunch of things, you are welcome to have the base removing the proton to make an enolate at the same time as the enolate attacks. But this arrow that we just showed with this arrow that where the enolate is attacking the carbonyl carbon, that's an addition step. This arrow that shows the base removing the proton, that's a proton transfer or deprotonation step. We are often okay with combining proton transfers in the same step as other things. Okay? So if we look at our drawing here, There is what used to be the nucleophile. In red is the bond, oops, I guess I'll highlight it like this, is the bond that we just made. In blue is what used to be the carbonyl carbon, and in pink is what used to be the carbonyl oxygen. Noticing that the two things that are attached, the two other things that are attached to that carbonyl carbon in blue, are those good leaving groups or not? They're CH3s. Is that a good leaving group or not? Nope, terrible. Terrible, terrible, terrible leaving group. So that means there's no elimination step that can happen. These are not good leaving groups. So there's no potential elimination step. The only thing you can do is to do a proton transfer step to get rid of the negative charge on the carbonyl oxygen. And you end up with this molecule where this used to be the carbonyl carbon, this used to be the carbonyl oxygen. Your two methyl groups from the original ketone are still there, and now you've made this new bond with what used to be the enolate. And that's an aldol reaction. It's a convenient way to make a bond between two sp3 hybridized carbons. One of the useful skills that can help you understand biochemical reactions is to recognize structural patterns in products that tell you a little bit about how the reaction happened. This product here we will call an aldol product, and aldol products have a key structural feature in common. If you start with the carbon that used to be the carbonyl carbon of the enolate, and then call this the alpha carbon, in an aldol product, the OH group is going to be at the beta carbon. So the structural pattern is aldol products are beta hydroxy carbonyl compounds. That is, they have a beta, they have an OH group, beta, to the carbonyl from your enolate. Yeah?
So when you're transferring the electron onto the carbon, when you bring the formal carbon, would you have to state it's like negative 3?
Oh, I think somebody should have raised their hand a long time ago. I'm missing an arrow, aren't I? What's the missing arrow? If you dump electrons into the carbon oxygen pi star, what happens to the pi bond? Breaks. Yeah, I broke the octet rule, which may be why you're worrying about charge.
Yeah.
Does that resolve the issue? Yep. Yep, thank you for that, and I'm sorry for that. There should have been protests. You should have been throwing things at me. Don't sit there and take this kind of stuff from a professor who you're paying to teach you, all right? Demand better. That seems like blaming the victim, which is bad. I'm sorry. But anyway, what I'm saying is, I'm sorry, that was a mistake. Minus 2 points for me. Yes? Yeah, so let's do the one, and that's actually also on your quiz. So let's do that one. Where is that? Let me just bring that in. So what I meant to do was I realized when I changed my mind in terms of how I was preparing the mechanism handout versus the quiz. When I started, I was like, okay, we'll do. Anyway, I decided ultimately to start with simpler examples on these new reactions and then go to difficult ones, and I never revised the mechanism handout accordingly. So what we'll see now for the aldol reaction is the one that's on your sheet. Let's just, I'll give you, oh, right, I want to put that in ability. So I will tell you that this molecule is the electrophile, and this molecule is the nucleophile. Now, because one of the important skills here is to focus in on what's important versus what's not, let's consider the electrophile first, and we'll go ahead.
On the products, are you missing carbonyl oxygens on the top?
Oh, brother. Yes, that is also a copy, paste, drag arrow. Thank you. This was not beta tested. That's horrifically embarrassing. Minus another 5 points for me. Yes, it was all part of my evil plan. Okay. Good, you identified something that didn't make any sense. Well, that's great. That's great, and that's you're starting to look at the starting materials and the products and like what's different, what's changed, and you were like, hold on, that doesn't make any sense. So that's great, and that's my mistake. I'm sorry. For the electrophile, we have 3 potential carbonyl carbons, and I will tell you 2 of them don't matter, and 1 of them does. Now, the fact that 2 of them are similar probably indicates to you which one matters, but why should I not worry about the carboxylate as an electrophile? What do I know about carboxylates? What level are they? 4, so not electrophiles, because they're already negatively charged. Now, how about a ketone? That's level 1. So it's no big, if you understand that and can go through one by one, even if you have no idea about what to do at the beginning, you can automatically dismiss the carboxylates as likely being spectators because they're level 4 reactivity, and then focus in on the ketone, which you know is a level 1 electrophile. And and since you know about ketones as electrophiles, you know a couple of things. First, there's no good leaving group on a ketone. Second, you know that the LUMO is the carbon oxygen pi star. So already, just by looking at this electrophile and the ketone, you should be expecting a nucleophile to attack. Even without knowing what the nucleophile is, you should be anticipating something like this going on. Okay, questions about how I drew that conclusion? Pretty clear? All right. Let's now go over here to the nucleophile. Now that's going to throw you for a loop because normally your nucleophiles have been things with lone pairs on them. And an OH minus or any of the other nucleophiles that we saw, something negatively charged. Here, we're dealing with a molecule that is neutral to begin with, but we're using a base. Because this is an aldol reaction, and you've seen the example, you know that you need to first deprotonate the alpha carbon. How do you know which is the alpha carbon? Well, this is the carbonyl carbon, this is the alpha carbon. You're going to deprotonate on the alpha carbon to make an enolate nucleophile. Now, if you didn't remember that, how would you tell what had happened? And the answer is, see if you can look at the products and see what has happened. If I look at that product from this immediate first step, I can pick up a couple of things. First, I can notice that what used to be my carbonyl oxygen is negatively charged. That gives you the idea that there was some kind of attack on the carbonyl carbon that led to a tetrahedral intermediate. Then I look at that carbonyl carbon and I ask, what was attached to it to begin with? And I see if I can find those in that intermediate. I sort of have 2 gray groups that used to be attached to the carbonyl carbon. They're still there, nothing changed. Here's the new thing. And when I notice that a carbon that is alpha to a carbonyl is bonded to this blue carbon, that's when I start to think that must have been an enolate that did the attacking. My nucleophile had to be an enolate, because that's the only thing I've ever seen where an alpha carbon ends up bonded to the blue carbon. So, how do I show deprotonating the enolate? From the base attacks this. If I wanted to, I could do this and then redraw the enolate separately. Alternatively, what do I do? Yep. Just have the electrons from the carbon-hydrogen bond attack the carbonyl carbon of my ketone directly. This arrow here is an addition step and describes the formation of a new sigma bond. That's this bond here in red between the alpha carbon and the blue carbonyl carbon. This arrow between base and the proton that's attached to the alpha carbon, that's a proton transfer arrow. So this is the stage at which you want to relax your eyes a little bit and see the similarity to things that you've seen before. Reactions work better when you're able to put them in your brain in sort of categories in related boxes, instead of seeing this as, oh my gosh, I have no idea what to even do with that. See this as nucleophile attacks carbonyl carbon and you break the pi bond. Just another example of that kind of reactivity. Yeah.
So I'm a little bit confused. So when the base attacks the hydrogen,
Yeah.
right? Wouldn't the bond go onto the carbon?
Yes.
So then how does that create the extra methyl?
Yeah. What we're saying by drawing it this way, drawing it the way I've drawn it here, is shorthand for the following. So what you're saying is you like that one more because it shows the electrons going onto the alpha carbon. And then the product of that is this molecule, which is an enolate, which is a nucleophile. And then you would show that enolate attacking the carbonyl carbon of the ketone. What we're doing here is just combining these two steps in one. Rather than show these electrons going onto that carbon to form a lone pair and then attacking, we're sort of showing that happening all at once.
I'm just confused on where the bond from the final stage come from, the red bond next to the.
Where does this one come from?
Yeah.
Comes from the electrons that used to be in this carbon-hydrogen bond. The hydrogen ends up on the base. Follow-up question? Others? Still no? Makes sense? Okay. By the way, don't feel pressure to nod your head and say something makes sense if it doesn't. We're here to learn, so if something doesn't make sense, let's talk about it. Yeah.
I'm just sort of confused how we get to that end result. Can you explain?
This end result?
Yeah.
Okay. This is a ketone. A ketone has a carbonyl carbon, a carbonyl oxygen, and two R groups that aren't leaving groups. That's why I've highlighted them in gray. And then you've seen before a nucleophile attack the pi star and break the pi bond. So fundamentally, that's exactly what we're doing here. The only thing that's different is the nucleophile. So this nucleophile starts out neutral. A base removes a proton from the alpha carbon. And when that happens, what you have, instead of your neutral molecule, you now have a negatively charged molecule. That is a nucleophile.
So, enolate, you're talking about that is the carbon?
The enolate carbon, the enolate alpha carbon is the one that attacks this carbon. We're trying to, later on it will be easy for us to talk about mechanisms in biological reactions if we don't show the proton transfer steps separately. It's just going to save us time. What I mean by what I've shown here is first base removes the proton, then you get a negatively charged enolate, then that attacks. But I'm combining the two steps to show them at once just to save time. It gets a little confusing because it looks like, wait, why did those electrons go there? You have to, it's just like when someone says y'all to you and you've never heard y'all before, it's you all, but it's contracted to be faster. This is a contraction of what's shown here. Yeah.
How do you know that the nucleophile attacks carbon 2 instead of carbon 1?
Great, yeah. How do you know that the nucleophile attacks, we're talking about this, 1, 2, 3, 4, carbon 2 instead of 1? Look at the functional groups. This carbon 1 is a carboxylate. It's negatively charged. That's level 4 reactivity, meaning not an electrophile. And it should make sense that a nucleophile shouldn't be that attracted to a carbon that's got a lot of negative charge near it, just because of charge repulsion. Same thing over here. This over here, carbon 4, is also a carboxylate. It's negatively charged. That's level 4, which means not an electrophile. In contrast, carbon 2 is a ketone. That's level 1. So we focus in on that ketone because it's simply more reactive than the two carboxylates.
So the nucleophile would have no reason to attack carboxylate.
Yep. Perfect. The nucleophile would have no reason at all to attack the carboxylates. Great. Good questions. Others? Okay, so that's the aldol reaction. You'll see this is a step straight out of the citric acid cycle. This is the first step in the citric acid cycle. This is how your body takes something called acetyl-CoA, puts it into a pathway that's ultimately going to allow you to exhale CO2 and generate ATP. Okay. We'll maybe have more to say about aldol reactions later. What I want to do now is talk about another reaction called conjugate addition, and this one's weird. All right? It needs a little bit of setup. Oh, great. Everything's weird. Needs just a little bit of setup. So conjugate addition is a reaction that's important in biology. It's a little bit different than typical carbonyl chemistry, but in some ways it follows the same principles. So I figured we'd talk about it here. I'm going to just redraw in my notes the molecule that's, I think, under that conjugate addition. We recognize easily the carbonyl carbon and the carbonyl oxygen. This is a ketone. And ketones are level 1 reactive electrophiles. You expect them to be electrophilic at the carbonyl carbon. This ketone is unusual in the sense that there is a pi bond adjacent to or conjugated with the carbonyl. The pi bond between the alpha carbon and the beta carbon is adjacent to or conjugated with the carbonyl group. When you have a pi bond adjacent to or conjugated with a carbonyl, you can draw a resonance structure where you push the electrons from the pi bond between beta and alpha carbons, you shift them over to form a new pi bond between alpha and carbonyl carbons. If you stop there, you'd have 5 bonds to the blue carbonyl carbon. So instead, you have to move the electrons between the pi bond from the involved in the carbonyl out onto the carbonyl oxygen. When you do that, you generate this resonance structure. The carbonyl oxygen is negatively charged, and the beta carbon is positively charged. Now that's an interesting observation, because if you remember what we learned at the very beginning of carbonyl chemistry, was that carbonyl reactivity in some ways is predicted by this resonance structure, where you move the pi bond out onto the carbonyl oxygen and you leave a positive charge on the carbonyl carbon. Do you remember that resonance structure we used earlier this term to talk about why carbonyls are electrophilic at the carbonyl carbon? So what that tells you is, the resonance tells you that the carbonyl carbon is electron poor. Similarly, resonance tells you that for this particular molecule that has this pi bond between alpha and beta carbons, we learned that the beta carbon is also electron poor. Okay? This type of molecule that has an alpha beta, C alpha to C beta pi bond conjugated with the carbonyl, we call this an alpha beta unsaturated, there's that word again, means with pi bonds, an alpha beta unsaturated ketone or carbonyl compound, whatever the case may be. So alpha beta unsaturated carbonyl compound. And what I've just tried to show you, and what your problem set also asks you to do, is to convince yourself using resonance that an alpha beta unsaturated carbonyl compound is electrophilic at the carbonyl carbon and at the beta carbon. 2 spots. Now, you're not going to know, looking at an alpha beta unsaturated carbonyl compound, you're not going to know how to predict whether a nucleophile prefers to attack at the carbonyl carbon or at the beta carbon. I don't expect you to be able to predict that. The way you'll be able to tell that it happened would be by looking at the products and seeing where the new bonds are. Are there questions about why the beta carbon shares the electron poorness with the carbonyl carbon? We're not really doing LUMO stuff here. We could. It's more complicated than I think you want to know. The conclusion is that the LUMO is on both the carbonyl carbon and the beta carbon. It's an extended LUMO, but we can leave that for another time, or perhaps not at all. Go ahead.
So we can treat that pi bond on the beta carbon as as a lone pair, right? Will we be able to do that in other similar compounds that aren't like a ketone?
So you've treated a pi bond in an alkene as a nucleophile, right? As the HOMO as a nucleophile, you've been able to use that in reactions. The adjacent ketone changes the chemistry of that pi bond, makes it much less nucleophilic. So we will see that alpha beta unsaturated carbonyl compounds are always electrophiles, not nucleophiles. So you actually won't be worried about what the HOMO is. If you had to, though, you would have picked the lone pairs on the oxygen over the pi bond anyway. Is that your question? Okay, good. Others? So there are two ways you could imagine a nucleophile interacting with an alpha beta unsaturated carbonyl compound. Two options for the nucleophilic addition step. The simplest would be to do just what you expect for carbonyl compounds. That is, your nucleophile attacks the carbon and you break the pi bond. Okay, that's attack at the carbonyl carbon. The second, and that would lead to an intermediate that looked like this. The second option is for the nucleophile to attack at the beta carbon. We've already said that the beta carbon is electron poor for reasons that were described above. If the nucleophile attacks the beta carbon, electrons in the pi bond between alpha and beta need to shift over, and you can draw that happening in one of two possible ways. You can just move them to be a lone pair on the alpha carbon, realizing that doing that, sorry, here's the nucleophile, doing that, putting the lone pair on the alpha carbon, results in resonance stabilization. You can subsequently take those lone pairs, move them over to form a pi bond between alpha and carbonyl carbon, and then break the carbon-oxygen pi bond with the lone pair out on the oxygen. The nucleophile, if it attacks the carbonyl carbon, ends up here. If it attacks the beta carbon, it ends up there. Two options. This one is called regular addition, or just addition. Attack at the beta carbon is called conjugate addition. So it's an addition step, but it occurs in a different spot than you expect. The reason it occurs at the beta carbon is because the pi bond between alpha and beta carbons is adjacent to or conjugated to the carbonyl. All right, so that's kind of a new reaction. The example here takes you through that. What you would need, and again, we're condensing via shorthand some proton transfer steps with some addition steps. You would have a base remove a proton from water. Then lone pair electrons are going to do the, on water, or on the oxygen, are going to do the addition step. You would need to recognize this as an alpha beta unsaturated carbonyl compound, which is electrophilic not only at the carbonyl carbon, but also at the beta carbon. Then your arrow comes here to the beta carbon, break the pi bond, move it on to the alpha carbon. That gives you this molecule. And then to get to the neutral product, you need one last proton transfer step here. Now you guys are not looking at the same example. What you're seeing on your mechanism handout is a little bit different, but look for the similar pattern, and I'll switch over to that in just a second. Questions about what I drew there? And this will be in my notes so that you'll have access to it after class. Here's the conjugate addition reaction in your mechanism handout. I'll should I erase what I drew and we'll go through it together. Okay. Blah, blah, blah. Here is, you recognize the need for a conjugate addition reaction if you can find an alpha beta unsaturated carbonyl compound. The other thing that tells you that this was a conjugate addition reaction is that the nucleophile is attached to the beta carbon. So look at where the nucleophile goes. This is actually a conjugate addition step from a process called beta oxidation. It's involved in fat metabolism. So whenever people like to talk about burning fat, and it's not actually combustion, it's this. This is what that means. Okay, so as we did in that previous example, we're going to use a base to deprotonate water to make it a good nucleophile. Then the oxygen nucleophile is going to attack the beta carbon. You can tell that it did because the OH group is attached to the beta carbon. We need, if we stop there, we'd have too many bonds to carbon, so we need to break the pi bond between beta and alpha carbons, move it on to the alpha carbon as a lone pair / negative charge. And then and that was here proton transfer followed by conjugate addition. We call it conjugate addition because it happened at the beta carbon. Then if you compare what we have here with the product, you can see that the alpha carbon used to be negatively charged and now it's not. So that stands to reason that the last step must be just a proton transfer reaction to get us to this product. The pattern for conjugate addition, you're always looking for the starting material to be an alpha beta unsaturated carbonyl compound, and you're always looking for the product to have the nucleophile at the beta carbon. All right, questions about that? I wouldn't have expected you to predict that, but knowing what you know now, I would expect you to be able to work your way through it and understand what's going on. We will talk about this reaction later when we do lipid metabolism. Okay, we need a break. There's 2 more reactions, then we're going to talk sugars. So let's take a few minutes and then we'll get going again.